objectives, now they're saying it's learning outcomes. So just to describe, I still use the old terminology, objectives. This is Dr. Manaz terminology, outcome. Describe how protein folding results in finite number of conformation. Also, we would like to identify these factors that could lead to denaturation of proteins. When I say denaturation of proteins, why your temperature is maintained at 37 degrees? Optimum for what? For the enzymes, right? If your temperature goes up to 42, you're causing protein misfolding. As simple as this. Acid-base balance disorder. If you have a problem with acid-base balance, you have, as a doctor, you have to treat that patient within a few hours, otherwise you're going to lose him.
I was at the nurse.
What do you mean nurse?
I had an allergy from the drugs. So I was at the nurse.
Okay, and the rest also have energy? Allergy? No. Just, you can get in, the others no. I made a rule, and I'm going to stick to it. If I'm late 5 minutes, you guys don't allow me to walk in. Please. So, this is the only way you can control. I mean, as I said, it is the male section, everyone's complaining about you guys. And I received it from all instructors as a year one director. It's unfortunate. That's why I'm strict about it. I'll be strict about it. Nothing else. If you're not interested, just be outside. It's fine. But don't, bother your colleagues by talking and so on. You're not interested? It's up to you. In the back, you're watching movies, whatever, do whatever you want to do. Here, you're here for the attendance. I could tell that. But at least do not disturb your colleagues. As simple as this. Let's be fair. And I'm telling you, I received complaints from all instructors about you guys. Female sides, wonderful. Male, please. Guys, I keep telling you, I mean, you're paying really hefty tuition that you need to get good education. Don't mess it up with these things. I mean, I'm trying to help people who are repeaters. Believe me, they have issues with attendance, with many of these issues, and that's why we're trying to resolve these issues. Try to pay attention. Believe me, if you listen, pay attention, it will allow you to understand better, but not only this, to shift from high school to college life. High school is different, college life is different. If you think that you're going to study on your own, good luck, but you're going to be exerting twice the effort if you were just to pay attention and listen to it. After all, you tried it before the TBL. Did it help? So try to do it another way. Now also, we're going to be talking about denaturation. We're talking about acid-base imbalances. We're talking about acid-base imbalances. Because if you change the pH of your body, you're causing protein misfolding. It doesn't take that much. I'm not talking about high temperature where you have to break or hydrolyze the amino acids in a polypeptide to break it down. You don't need that. Just small change and the protein, you have completely different non-functional protein. As simple as this, that's not compatible with life. So this is why protein misfolding is very important. We're going to talk about Alzheimer's, we're talking about so many diseases, all caused by protein misfolding. And you need to know the mechanism of these conditions. We will talk about quaternary structure. Hemoglobin. How many polypeptides we have for hemoglobin? 4. Myoglobin? 1. Which one has quaternary structure? Hemoglobin. What about myoglobin? Tertiary structure. So hemoglobin, because the fact that it has multiple subunits, has quaternary structure. And that also plays a role in terms of the protein misfolding. Now we will discuss also some of these protein misfolding. The common one is the Alzheimer, Creutzfeldt-Jakob disease, which is the most common one for prions that we will discuss. And this one actually is the same, prions causing the mad cow disease. But also I'm going to show you that can proteins be infectious? We always talk about viruses. We always talk about bacteria, right? Can protein infect another person? Yes. When you eat mad cow disease, infected cows, you can contract it. But you just had proteins. So are you telling me if I eat? Yes. If you transplant organs from one patient with certain conditions that have prions, the patient, the acceptor, will contract that condition. I'm talking about proteins being infectious. I'm sure that this is the first time you hear about that. Well, it's a fact. Do we have prions in our bodies normally? Prions, yes. The infectious protein. We don't have it, but we have prions, the normal ones. But then they change, cause protein misfolding. Now they become infectious, and they cause the diseases you're going to talk about, including the mad cow disease. Okay? And then we're going to talk about proteins resulting from mutations, such as cystic fibrosis and sickle cell anemia, that could change the protein misfolding or reduce protein misfolding. Here are nice pictures. I know you just wonder how difficult it is. It's not that difficult. Now we have software that can do it. If you feed the sequence, it will simulate it for you. And we're talking about hemoglobin. This is the 3D structure. Why do we need to know these? If you were to design a drug targeting these proteins that could misfold and so on, you have to know their structures. This is why we have this folding structure, 3D structure. I took crystallography, where we utilize crystals to come up with the 3D structures. I took it with Hartman Woodward, who won the Nobel Prize for his formulas to come up with these structures. Of course, if you're complaining about me, I would love for you to take a lecture with this guy. And when you understand something, you just come and explain it to me. The only thing I understood from this course is basically I enjoyed the chicken wings that he invited me to take at the end of the course, because he invited all the students. I'm talking about crystallography. It's really tough. My crystallography, other professor for crystallography, he had a tie with insulin structure that he came up with because it was done in the space. Because crystals can form nicely in the space, and they can come up with better resolution for the crystals. Why do they do it in pharmaceutical companies? Because they want to design drugs. They could say, okay, if we target this region, how can we come up with, you can model the drug to bind that region of the protein. This is why this is crucial to designing drugs. And they have so many softwares to modulate these things. Just wanted to introduce it to you. But the thing is also, it's a dynamic process, protein folding, meaning that proteins just not, you don't have the folded protein as static. It changes with the pH, with the temperature. It keeps changing. So you can see it's the same protein, just having different folding. Okay? So if we know the structure of this protein, we know most likely the function. We can predict the function, whether it's a fibrous protein, whether it's an enzyme. From the structure, we can figure out the structure of that protein. So also, in terms of evolution, like for instance, I'm working on sirtuins, the anti-aging proteins, because we found that in the prokaryotes, Drosophila, and the Candida albicans, when you upregulate this protein, you double the life of these organisms. So what is the equivalent protein that we as humans have it compared to this SIR2, called SIR2, SIR2? We came up with 7 proteins that share similar structures, and now we're trying to figure out which protein is involved. So it's good to know about these structure of the proteins, design drugs, and protein problem folding. So we know what's the protein folding, but also proteins, as I said, they're not static. They bind ligands, such as enzymes, they bind substrates, right? So what do they do with the enzymes? Either they can lock model, you just bind to it, or they could induce the conformation, the folding of the protein, so that the substrate or the ligand could bind the enzyme, the protein in this case, right? So the question is, when this binding occurs, what happens? The conformation changes and allows other factors to bind. Hemoglobin. Did you study about cooperative binding of oxygen? Meaning that when oxygen binds, The first one, it changes the conformation, the 3D folding of the protein, allowing oxygen to easily bind in an easier manner. So this is what we call cooperative binding. One binding changes the conformation, then the second one binds easier and easier and easier. So this 3D conformation occurred to assist or facilitate the binding of the ligands. But also, we talked about transcription factors, right? We bind DNA, changes the conformation, allowing the other transcription factors to bind and form the complex to initiate transcription. So there are changes in the conformation. Now, these come from the fact that the side chains of the amino acid determine how polypeptide chain folds. I'm going to give you examples, such as sickle cell anemia, where glutamic acid gets changed to valine. Glutamic acid is acidic acid. Valine nonpolar, it's a side chain, resulting in completely a different protein that precipitates, forming the sickle cell syndrome, that, as you will see. These side chains do affect the 3D structure. It is done random process, and also the thing is, a fully folded protein is the native functional form of the protein. So when it's fully folded. Now, denaturation of proteins, when I say denaturation, as I said, we're not breaking down the polypeptide links. We're just changing the 3D folding, which doesn't require that much. So denaturation basically could affect the activity of the protein. So without the hydrolysis of the peptide bonds, denaturing agents include heat, organic solvents, strong acids or bases, detergents. Why these could kill you? Because they use denaturation of the protein. Not only this, most proteins, once denatured, remain permanently disordered. They lose function. You lose them. You have to get rid of them. Your body gets rid of them. And then they precipitate from the solution. So when they denature, basically they're exposing the hydrophobic sides that are usually covered in the protein. They get exposed. As a result, they precipitate and aggregate, and this is how they cause damage in many of the conditions and diseases, as you will see. So here's just an example of denaturation for a protein. You can see what we call melting temperature of the protein, which is the temperature required for 50% of the protein to unfold. And usually it ranges from 40 to 60 degrees. This is why you take the temperature above than 42, 43 in a human being, it's incompatible with life. Because proteins start getting denatured. You don't have to denature all the proteins. Some will be enough to basically kill you. That's why it's crucial to maintain the temperature at 37 degrees. Except tag DNA polymerase that we use for PCR. We use it at 95 degrees. Have you wondered how could it be that it's a protein? Well, this was isolated from the Thermus aquaticus bacteria. This was isolated from the Yellowstone National Park hot spring. I mean, there's bacteria growing there at 95 degrees because it has proteins that could tolerate. Okay, and this is how Karen Mullis, anyone knows about him? The guy who invented the PCR. Basically, he just used this tag polymerase. It was 1964 discovered, and then just brought 2 water baths, and he made billions of dollars using this protein. Anyway, so we have chaperones. I'm sure that you heard about chaperones or heat shock proteins. Anyone wondered what's heat shock proteins?
Chaperones is proteins that help other proteins to do its function.
No, not exactly.
To fold the proteins.
Fold properly. It helps them to do their function. How? By helping their folding, proper folding. Okay? So these are what we call chaperones. Another term, another name for it, heat shock protein. I'm sure you're going to hear about heat shock protein, 70s and so on. Why do we call them heat shock protein? Because heat, what does it do to these proteins? It induces them and affects the folding of the proteins. But also, we have chaperone. This is an example of chaperone or chaperoning. What's the difference? Chaperonin. Chaperonin is a class of chaperone, portion of it. Chaperone is a general term. Proteins could be small proteins, a symbol around the unfolded protein, and then they induce its folding, proper folding, and then they dissociate. However, chaperonin, this is example of chaperonin, small proteins can get in with the utilization of ATP, then this protein gets folded properly, and then it gets ejected or expelled from the, in this case, the chaperonin. So chaperonin is a class. I mean, it could be small proteins. This is all explained to you in the movie. I'll let you watch the movie yourself. I don't have time for these, but this could explain to you the whole process of chaperones. How they, basically, if I ask what's the function of chaperones, they induce proper folding, protein folding, right? If there is something wrong with it, now there's a big project we're doing in the National Guard, where we're sampling patients from Al-Hajj who have heat shock and their effect on the protein folding by checking the heat shock proteins and so on. So there's a lot of research interest in terms of the folding on people who come during the Hajj season. Now, quaternary structures, we already talked about it. I'm not going to repeat it, but just want to show you examples of hemoglobin, 2 alpha subunits, 2 beta subunits. These are the heme with the iron molecule inside. This is collagen, the most common protein, the connective tissue. You can see it's composed of 3 polypeptides called like a roll. Okay, so this is quaternary structure. This is the myoglobin. Quaternary or tertiary? Tertiary structure. So protein misfolding. The problem with misfolding is the formation of aggregates. I told you that when a protein misfolds, what happened to the hydrophobic regions? They expose. They become insoluble. This is similar to the oil. Hydrophobic, it doesn't mix with water, right? So the same thing with the protein. Hydrophobic, insoluble, precipitate, aggregate, and that's what kills our neurons in most of the diseases. That's the whole idea. This protein is folded that could occur because of temperature, because of acidity, because of acid-base imbalance, toxins. All these could induce protein misfolding. We don't need that much to break down the peptide linkages between amino acids. Just simple changes, and then you lose the function, and that's permanent in most of the protein. The protein misfolding, we're going to take examples of DNA mutation, cystic fibrosis. We're going to talk about also sickle cell anemia. Stress could also lead to protein misfolding. Environmental factors, metals, pesticides, toxins. You know, when people are spraying pesticides into the lawn or grass, you tell the kids don't play on it, right? Because it could penetrate through the skin easily. So this is why you don't need that much to kill the kid, because it can really penetrate, and all what it does, most of these toxins, just basically induce protein misfolding, as simple as this. That's why it's crucial to understand protein misfolding. So the whole idea is to expose the hydrophobic segments to water, which is generally unfavorable. As a result, we do have the aggregates that are highly stable, and once aggregated proteins are too large, they become insoluble and fall out of solution. And this is the problem with these aggregates. So let's talk about amyloid diseases. You, as a doctor now, you're going to try to link the protein misfolding to so many conditions. Amyloid. If you mention amyloid, you always think of Alzheimer, right? So we do have several neurodegenerative diseases that you will study during your course of training about neurodegenerative diseases such as Huntington's, Parkinson's, and Alzheimer's diseases. These are all caused because of protein misfolding. As simple as this. This is an example of amyloid fibrils. If you stain for it, this is Alzheimer. nervous tissue, you can see that they're accumulating. This accumulation of these fibrils, amyloid fibrils, kills your neurons. As simple as this.
So why is Alzheimer only common in old age?
Because it takes time for these proteins to accumulate. That's all. It's happening in our bodies. Our bodies, because of the immune system, can take care of it and can basically get rid of it. But as we get older, my belief right now, most of the diseases that we have are all immune related. We have cells in the nervous system that's capable of taking care of it, similar to the monocytes. Which cells? Astrocytes. And also, what about the microglial cells? Any monocyte equivalent will take care of it. When these cells, modulated by the immune system, stop functioning, you have the accumulation right there. You see what I'm saying? So that's why I really think that most of the diseases are caused by immune system. If you maintain a good immune system, you'll protect it, including cancer cells. If we have cancer cells, your cells, cells, could kill it. But if there's a problem with the immune system, will not kill it. And that's where the problem is. So most of the diseases, you play with the immune system. I'll tell you an example, Hashimoto's disease. My daughter was diagnosed with Hashimoto's disease. Of course, we panicked, blah, 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 blah. That was 10 years ago. Now, what I did, I have my brother-in-law who is a pharmacist, and he said he treats the immune system with different supplements, vitamins, herbs, and so on. I said, can you help? And he did actually give us a lot of extracts, coconut extract, vitamins, blah, blah, blah. And he said, use it, around 5 bottles of different extracts. And he said, this will play with the immune system. We gave it to her for 4 or 5 months, and since then she's free. As simple as this. He played with the immune system. Eczema, immune system. Most of the diseases, diabetes, immune system, whether type 1 or type 2. And we'll explain to you about type 2, even type 2 with aging, immune system related. It's your white blood cells, in particular monocytes, that are causing a lot of these diseases. This is why. They stop functioning, they stop taking care of these precipitates or aggregates because they're supposed to engulf them. You don't do that, they accumulate, they kill the nerves.
What does this disease do?
Which disease? Hashimoto's disease? Thyroid, or the thyroid. Kills your thyroid gland. So you need to take thyroid supplements for the rest of your life. It's an autoimmune reaction. We'll talk about it. Okay? What I'm saying is that take care of the immune system. Then you'll take care of a lot of diseases. You'll find out. And believe me, this is my experience with so many research projects. The immune system is the one that causes insulin resistance. If you talk about diabetes type 2, that's all mediated by immune system monocytes, the same thing. So, here are some amyloid fibril formation diseases, basically resulting in Alzheimer's disease, Parkinson's disease. This is just the normal protein. When it misfolds, you have aggregates. This could lead to prion diseases. We discussed the infectious prion. I'm going to describe it in more detail. Huntington's disease, but believe it or not, now there's research towards type 2 diabetes that amyloid